Hello viewers. Previously we discussed about IR spectrophotometer and etc. Therefore, to know the concentration or to know structures etc. But what about the reaction time? So to know that how much time taken to complete a reaction, just we are discussing now about thin layer chromatography. Okay, let's see the basic things required for chromatography, that is thin layer chromatography. We need a TLC plate, usually made up of glass or aluminum or some other materials, and we need a solvent and a sample to separate, and capillary tubes and developing chamber, that is glass or plastic container, and spotting guide or template, a pencil, and a UV lamp for those whose drops are not visible, that is those points are not visible, then we use UV lamp or visualization sprays etc, and a roller or measuring tape. Okay, let's take a chromatography paper. It is available in your lab. Don't use other papers because chromatography paper is different from others. It is made for chromatography. And take a silica powder and made it like a paste using methanol or something solvents that are suitable for the reaction or chromatography. Using a roller and pencil, draw a line of 1 centimeter from the below line. Take the solvent and add little amount to silicon and make it as a paste and spread it over the chromatographic paper. And now we will go for the chromatographic reaction. That means what reaction we need to observe. Let's take any reaction. Please take a small type reaction, that will last long within hour or half an hour, because we need to observe until we the reaction complete. So take less time reactions. Example, NaCl, or but one thing to remember is take polar compounds for polar solvents. For non-polar compounds, take non-polar solvents. Take drops of initial reactions with the help of capillary tube and add it to the initial line of chromatographic paper that is drawn before, but with equidistance, and then place it in a chamber, that is chromatographic chamber or glass jar, and place the lid or cap, but uh, don't make the line that drawn into the water. The water level must be below the line. After some time, remove the chromatographic paper and observe it. If it visible, then okay. Else, you use sprayers or UV light to observe the spots. If the spots move from baseline to above the baseline, then the reaction started. It means, or if it doesn't move any further, it means it not started at. That means the reaction didn't started yet. Okay, after some time, do the same process for again and again until the baseline spot disappears. It means the reaction completely. After some time, remove the chromatographic paper and observe it. If it visible, then okay. Else, you use sprayers or UV light to observe the spots. If the spots move from baseline to above the baseline, then the reaction started. It means, or if it doesn't move any further, it means it not started at. That means the reaction didn't started yet. Okay, after some time, do the same process for again and again until the baseline spot disappears. It means the reaction completely